Julia completed her postdoc training at the Weizmann Institute of Science and Technology, and she recently joined the Bordeaux Institute of Oncology, where she's investigating immune system aging in clinical contexts with a particular focus in oncology. So today we will hear about her research that she mainly did in Israel, and we are looking forward to your talk. Thank you very much for accepting our invitation.
Thank you so much, Anna. Thank you so much for your kind invitation and introduction. So today I will tell you, today I will focus on aging. I would like to listen about aging and in particular about senescent cells and how do they trick immune system to escape immune-mediated clearance and promote age-related lung inflammation. Okay. So I would like to start from this simple picture of two old mice, which are litter mate. They're 2 years old, but one of them was treated with a drug that induces apoptosis of P16 senescent cells, the mice on the right, and on the left is just a control. As we can see from the first side, that mice with depletion of P16 positive senescent cells looks way better. And this is not only the way the mice looks, careful investigation revealed that functioning of a wide array of organs in the mice with depletion of senescent cells that was improved. Moreover, these mice lived longer compared to the control. So this seminal work from by Darren Baker in 2016 put senescent cells in the spotlight and attracted lots of attention because as senescent cells were known to be associated with aging, it was shown actually in the seminal work for the first time that they are drivers of aging and age-related diseases, and their elimination is actually extending health span and lifespan. So let me start from the beginning. What are these cells? What are senescent cells? Senescent cells, the simple answer would be that they are, upon some kind of damage, they entered irreversible growth arrest. But biology is not that simple and black and white. So we already have the understanding that cellular senescence is a complex cellular state. And senescent cells in vivo can be heterogeneous, playing beneficial and deleterious role, and it really depends on the context. However, to identify and characterize them, we know that there are some common feelers that helps to identify them. And so, as I mentioned already, upon stress, and it's usually DNA damage, but it can be oxidative stress, oncogene expression, cell fusion. So upon stress, there is activation of DNA damage response and upregulation of gamma H2AX protein. But most importantly, what these cells upregulate, they upregulate inhibitors of cyclin-dependent kinases, which are indispensable for cell cycle progression. And these are so-called molecular markers or identifiers of cell senescent state, being P16 and P21 in particular. Moreover, these cells are resistant to apoptosis. They elevate proteins from BCL family, which increases the resistance to apoptosis, and they are metabolically active. They secrete a vast number of cytokines, chemokines, also metalloproteases that can modify the niche they reside in. And this is under the umbrella of senescence-associated secretory phenotype. And these cytokines that they secrete, they can also call immune system to come and interact, and since senescent cells are immunogenic, can be removed by immune cells in vivo in immune surveillance process. So as I mentioned, cellular senescence is complex, and also its role in vivo context will depend on their persistence in the tissue. And we already have, I would say, good understanding that short-term cellular senescence in vivo can be beneficial, as they play active role in tissue patterning, in embryonic development. They also, in acute injury, they help wound healing. And when you think about this, that they suppress aberrant proliferation of damaged cells, is very good. Tumor suppression function. However, when they are persistently accumulating in tissues, they are deleterious. And their long-term presence can lead to tumorigenesis and other age-related diseases. And so we can, how do we intervene with their accumulation? We already know that we can target the proteins which are upregulated from BCL family and that give resistance to apoptosis. And these drugs are so-called senolytic drugs, which aim to clear senescent cells. However, here the problem is that these drugs are not only specific to senescent cells, and they have side effects that can be toxic in vivo. So other arm is immune-mediated surveillance, biologically relevant. However, in aging, things go aberrant and it doesn't function well. So I wanted to concentrate exactly on this arm, how these cells are eliminated, or actually what happens that they are not eliminated, and what contributes to their persistence in the tissue. So when we think about the accumulation of these cells in vivo context, we can put it as a simple equation, where we would have DNA damage as a factor inducing production of these cells in vivo context and their removal, right? That would be immune-mediated clearance. However, with age, there's more DNA damage load, so there will be higher production rate, and removal is worse. We know that the immune system doesn't work in old organism as it used to in young. It's impaired. However, what was not known was whether senescent cells themselves can regulate immune system activity and their own removal by intrinsic mechanism. And I wanted to really understand this crosstalk between senescent state and the immune system, and whether we can target and whether the consequences would be beneficial. So I wanted to understand this phenomenon at single cell level, and I used lung as a model organ in physiological aging and chronic lung inflammation, that is age-related state. And I wanted to understand which cells undergo senescence and how do they interact with immune system. So I was interested in their immunoregulatory phenotype. And for vast majority of my experimental system, I use mass cytometry, because at the time when I started it, it was the only way to actually measure it, high throughput, many markers in parallel, where I could characterize different lineages and functional state at single cell level using protein-based methodology. So I would like to start from the first intriguing result that relates to immune system, and in particular myeloid lineage and alveolar macrophages especially, which I found that in aged mice are overexpressing senescence-associated markers, and these are inhibitors of cell cycle progression, inflammatory markers, and the resistance to apoptosis. And same population was very highly upregulating immune checkpoint PDL1 protein. PDL1 is programmed cell death ligand, and this is central regulator of immune responses. And this drew our attention whether these two factors are linked together. And indeed, cells, alveolar macrophages, which were highly expressing P16, they were also highly upregulating PDL1 protein level. And this was in both age groups, so regardless of age. However, it was more pronounced in older mice and at population level and also at single cell level. And this phenomenon was not reserved to particular cell type, alveolar macrophages, but I found it in different lineages. Like here I'm showing you lung epithelium, same phenomenon, but it was also, but also in chronic lung inflammation. So different experimental model system which I investigated was same. So cells simply which were high P16 positive senescent cells were highly upregulating PDL1 level. And this was really intriguing, and moreover, like, what are these cells, whether they can be found in clinical context. Indeed, yes. I observed that in patient-derived samples with lung pathologies, we could observe accumulation of the cells in emphysema, but a lot of them, in particularly in different cancer types of cancer, lung cancer. So this leads to the question whether there's molecular mechanism of PDL1 upregulation in senescent cells. And to address this question, I referred to well-established in vitro cell culture system, where we can induce senescence either with DNA damage-inducing agents or by extensive replication. And in both of these model systems, it's well established that these cells are expressing inhibitors of cell cycle progression, like P16, and they were also both upregulating PDL1. This is level of PDL1, sorry that this is not marked. Moreover, when I overexpressed P16 on its own in growing cells, PDL1 level went up. And when I used, in addition, in different settings, when I used pharmacological drugs simply that are FDA approved for oncology patients, palbociclib or abemaciclib, they pharmacologically they mimic P16. They bind to CDK cyclin-dependent kinases for 6, and they arrest cell proliferation. So when I use these drugs on growing cells, PDL1 level went significantly up. So this all suggests that indeed upregulation of PDL1 checkpoint in senescent cells is mediated by P16 dependent inhibition of cyclin dependent kinases. But what happens next? So in other model system, I investigated what would happen if, in contrary, we would knock down P16 gene encoding P16. And so I took senescent cells, which have initially high level of PDL1, and I knocked P16. And what I observed is that PDL1 level went down. Moreover, when I thought that maybe the turnover of PDL1 protein changes, and this was actually when cells were treated with a drug that stopped translation. So when I can follow lifespan of the given protein, and here PDL1. So senescent cells with knockdown of P16 had higher rate turnover rate of PDL1. And this really suggested that senescent cell upregulate or increase stability of PDL1. And PDL1 is known to be, is known to be ubiquitinated for subsequent degradation in proteosomes. So but there are many E3 ligases. So I followed, like, there's in literature, it's already known that which ligases can be corresponding one that are in charge of turnover of a given proteins. And since PDL1 is well studied in the context of cancer, I followed on a given E3 ligase complex that was known to be regulating PDL1 protein level, its ubiquitination. And so this is very complex machinery composed of many proteins. And one of the proteins, so-called adapter protein, here in this complex is called the beta-TRCP. And what does it do? It simply recognizes substrate proteins like PDL1, binds them, and targets them to this complex for ubiquitination, subsequent degradation. And what they've seen is that senescent cells actually had lower level of adapter protein compared to the growing control, meaning or suggesting that it is very likely that in senescent cells, PDL1 is not as efficiently targeted to the E3 complex for ubiquitination and degradation, and indeed ubiquitination level was also in senescent cells was also lowered compared to the growing. So all of these results support molecular mechanism behind upregulated levels of immune checkpoint protein in senescent state. Simply by inhibiting CDK proteins and upregulating its stability by downregulating the turnover rate. So PDL1 is a well-studied protein in the context of cancer. Cancer cells often elevate this protein in order to suppress immune arm, in order to suppress T lymphocytes or lymphocytes, not only T cells, but can be other lymphocytes. And what does it do? Simply PDL1, upon interaction with its receptor present on lymphocytes, PD1 receptor, it can induce state of anergy of lymphocytes, even induces apoptosis. It simply suppresses immune arm. And this is the way how cancer cells can evade immune-mediated clearance. And here, what we thought is that senescent cells simply do the same. They elevate the immune checkpoint in order to protect them and escape from immune-mediated clearance by suppressing immunity against senescent cells. So, but there are means to tackle this problem. So cancer patients are treated with immune checkpoints inhibitors, and simply what does it do? You have to inhibit this axis, PDL1 interaction with PD1, and this is how to stimulate. This is what is done in cancer patients to stimulate immunity to kill cancer cells, and this is what we've done. I used the anti-PDL1 antibody, which not only inhibits the interaction between PDL1 and PD1, but it was activating antibody, meaning that the Fc part of this antibody has high affinity towards Fc gamma receptor positive cells. And these are NK cells, macrophages, neutrophils, monocytes, and it stimulates their cytotoxic activity. So simply it does two things in one. It blocks the axis, PDL1, PD1 axis, and also stimulates cytotoxic activity of Fc gamma receptor positive cells. And I checked this approach I checked this approach in short-term models, so whether we can implement such strategy, treat mice with anti-PDL1 treatment to eliminate P16 positive cells. First, I've done it in short-term damage model to the lung. Mice were exposed to LPS, it causing inflammation and elevation of P16 PDL1 positive cells. But upon the treatment of these mice, I've seen that this simple treatment, which lasts like twice injecting an antibody, the drug, that leads to significant depletion of P16 PDL1 positive cells out of general immune compartment and also alveolar macrophages. This was well corresponding to activation of cytotoxic or activation of immune mart. Here I'm showing you T lymphocytes in particular. So same approach I applied in long-term model in aging mice. So I took young and old mice, and old mice were treated either with isotype control or anti-PDL1 antibody. And I checked the level of senescent cells, and it proved to be successful. Of course, here the treatment was longer. It lasted 3 weeks. What we observed upon such treatment, there was depletion of P16 and P16 PD1 positive cells, again from general immune compartment and also alveolar macrophages, and it corresponded well with it was correlated well with the activation of T cell response. So PDL1 treated mice had activation of cytotoxic CD8 T cells. And also was important, there was reduction of inflammation, which I measured in the blood. So systemic inflammation of these mice was reduced. Same approach I implemented in long-term damage, chronic inflammation. So mice with chronic inflammation, they were treated same again with either isotype control and PDL1 antibody, and it proved to be successful in depleting P16 positive cells and also P16 PDL1 positive cells out of immune compartment general, or also here closer look at alveolar macrophages. It corresponded also well with general decrease of senescence-associated markers in lung tissue and reduction of inflammation. Just as a small bonus, I compared two treatments of PD1, anti-PD1 antibody and anti-PDL1. We were asked by reviewers, but it was also very interesting for us what would happen if we would target other arm, so receptor that's present on lymphocytes, and actually it didn't prove to be successful. Anti-PD1 treatment couldn't deplete P16 positive cells. So this leads us to conclusion, to summary slide, where I described you the model that follows a particular scheme, so we can think about accumulation of senescent cells in this context, that we have a damage that leads to induction of senescent state by upregulation of cell cycle inhibitors. Here I put main focus on P16 inhibitor of cyclin-dependent kinases. It leads to downregulation of ubiquitination of PDL1, and this leads to increased stability of the protein and accumulation in senescent cell. Upregulation of PDL1 can lead to suppression of immune response, and persistence of senescent cells in vivo in tissues, which leads to overall increase of chronic inflammation and senescence-mediated age-associated diseases. However, we can intervene by targeting PDL1 by anti-PDL1 antibody. Here I use activating PDL1 antibody, which not only inhibits the axis between PDL1 and PD1, but also stimulates activity of Fc gamma receptor positive cells, and it leads to depletion of senescent cells and decrease of general inflammation, and it can open future ways to design strategies which would be anti-aging based on this approach, based on anti-PDL1 antibody or targeting PDL1 antibody. I would like to thank all of you for listening, and especially people who are involved in this research in Weizmann Institute. It was really a pleasure to work with them together and to simply come to some good, promising conclusions. I'm very happy about this and to publish this story. And I'm very happy to take any question. So